This is a production of Cornell University.
Thank you much for the introduction. So first, I will give a quick a few background slides for my talk, and then I will proceed to introduce our recent finding in the involvement of aquaporin in aluminum resistance and heavy metal detoxification. Yeah, finally, I will give a quick summary for some of my recent ongoing project in the lab. So introduction. Plant requires essential nutrients for their survival and growth. The essential nutrients can be classified into two groups: the basic non-mineral and mineral nutrients. The basic non-mineral nutrients are supplied to plants through air and water. You can see carbon, hydrogen, and oxygen. And while the mineral nutrients come from the soil. Mineral nutrients can be classified into macros and micronutrients. There are 6 macro and 7 micronutrients. So you see the micronutrients, many of them are transition metals, so that they have a very narrow dynamic range between requirement and toxicity. So for instance, the copper, iron, magnesium, and zinc, they all transition metals. There are also metals and metalloids with no structural and cellular functions, but they can be toxic to plants under certain environmental conditions. For instance, like aluminum, cadmium, arsenic, etc. The soil provides plants with essential nutrients. However, plants can encounter soil-based abiotic stresses. These include drought, flooding, mineral deficiency, high salinity, or heavy metal toxicities, adverse pH conditions, either too low or too high. So all these abiotic stresses can inhibit plant growth and development, leading to yield losses. So my laboratory research is focused on heavy metal toxicity and low pH or acidic soil related stresses, including low pH stresses and the aluminum stresses associated with the low pH. For non-accumulating plants, they take 2 general strategies to cope with metal toxicity. The first strategy is an exclusion mechanism. With this mechanism, plant roots release organic compounds to the rhizosphere, forming non-toxic complex with the toxic ions in the rhizosphere, which cannot enter the root cell. So that's how the exclusion happened. The second strategy is the so-called internal mechanisms, which deals with toxic metal ions in the plants. In general, plants move the toxic ion away from the most sensitive part of the plants, or sequester the toxic metal ion into the vacuole of the root cells or leaf cell or above ground of the above ground tissue. Also, different metal ion toxicity have different target. For instance, aluminum toxicity target at the root, especially the root tip region. So in contrast, zinc toxicity targets at the shoe, like a leaf. So the second section, the involvement of aquaporin in aluminum resistance and heavy metal toxicity. So first, I would like to introduce aquaporin. Aquaporin, another name is water channel. So it constitutes a large and evolutionarily conserved family in plants. For instance, there are 35 members in Arabidopsis. Based on their subcellular localization and function, the members can be further classified into at least 4 subfamilies, so including the plasma membrane, intrinsic protein or PIPs, tonoplast, intrinsic protein, TIPs, nodularly 26-like intrinsic protein, NIPs, and small basic intrinsic protein, SIPs. Aquaporins are channel proteins that facilitate the passive transport of small molecules across various cellular membranes. It is generally believed that aquaporins facilitates polar but facilitate the transport of polar but non-charged small solids, so such as water, carbon dioxide, ammonia, arsenic, boric acid, etc. So it's still an argument whether aquaporin can transport charged metal ions. So I will talk about more on this issue. So in my next talk. So first, aquaporin and aluminum resistance. First, I will introduce what is aluminum toxicity. Aluminum toxicity occurs when the soil pH is below 5.0, or I mean, it's acidic soil. Under this condition, the toxic aluminum ions, or Al3+, is released from the soil particles, which is highly toxic to the root, especially to the root tip region. So here shows 2 wheat lines, one is tolerant Karasenko and the sensitive aggregate. So you can see under the neutral soil condition, both are defined, they are healthy, no difference. But under acidic soil condition, you can see the root growth was greatly inhibited by the acidic soil. So this inhibited root growth limits water and nutrition uptake. So that's the reason, I mean, the plant, you see the stunting plant and loss of yield. So in Arabidopsis, the plants mainly take the exclusion mechanisms to cope with aluminum toxicity, so which is achieved by root release of malic acid and citric acid from the roots. And this process—
Can you move the bar?
So the bar, okay.
They're blocking.
Okay, yeah, sure. Yeah, thank you. Yeah, so these are 2 transporters. ALMT1 stands for aluminum malate transporter. It's responsible for malate release from the root tip. And AT-mate is the citric transporter. It's responsible for the release of citric acid from the more mature shoot, the root region. So the question is whether an internal detoxification mechanism is involved remains unclear in Arabidopsis. So the next, how was aquaporin linked to aluminum transport and resistance? So before I proceed, I would like to introduce the concept of aluminum accumulator. So in nature, most of the plant species, including crop species, are so-called non-accumulating plants, which means they cannot tolerate high concentration of aluminum or other toxic metal ions in their above ground tissues. But there's a small number of plant species, including crop species, they can accumulate high levels of aluminum in their above ground tissues without showing any toxicity symptoms. So this includes several quite known plant species, like tea is a accumulator, buckwheat also, and flower, the hydrangea. So the hydrangea plants are interesting. Okay. So in that they have red and blue colored flowers. Amazingly, the color difference is not depends by genetic variation, but by environmental factors. So specifically, it's the soil pH. So under neutral soil pH condition, the plant produce red flower. But if you switch the plants to low pH or acid soil, the flower color turns from red to blue. So just like a magic. So how can I don't know this. Let's see. This one, no? What should I? Okay. Yeah.
Should you go back?
Yeah, that's fine. Yeah, that's fine. So I've just finished. So how can pH change the color of hydrangea flower? The answer is aluminum. So under the neutral pH soils, the flower shows the native red color of the anthocyanin stored in the vacuole of the sepals. So what we see here is actually sepal, but not petal of other common flowers. So under acidic soil condition, aluminum is released from the soil and taken up by plants and translocated to the above ground tissue and finally sequestered into the vacuole of the sepal cell. So the aluminum there will form a new complex with the anthocyanin, turning the flower, the new complex, it's blue color. So this is the secret for the magic. So Dr. Kumi Yoshida's lab in Japan, they are working for the horticultural trade and they study the mechanisms underlying the flower color change in hydrangea. So using differential expression approach, they find two genes that encode proteins that are responsible for aluminum uptake and color change in the sepal cells. So these two proteins, one is called PORT, so stands for plasma membrane aluminum transporter, and WAT stands for vacuolar aluminum transporter. So these transporters are responsible for taking up aluminum across the plasma membrane into the cytosol and further sequester into the vacuolar. Both PORT1 and WAT1 are members of the Aquaporin gene family in hydrangea. So for us working on aluminum resistance, so the natural question is, could Aquaporin member be involved in aluminum resistance in plants? So to address this question, we did a phylogenetic analysis and found out that the NIPS members are the closest homologue of the POT1 gene in Hydrangea. So next we examine the phenotype of the individual for individual members, their mutants, and see their performance under aluminum stresses. So that's what the summary. I mean, we saw that the NIP1-2 mutants are more sensitive to aluminum toxicity. So under the standard condition, root growth of the wild type and the NIP1-2 mutant cannot be distinguished. So that means this gene is not essential for normal function. But under aluminum stresses, we can see the more growth inhibition in the mutant compared with the wild type. So this result indicates the wild type function of NIP1-2 is involved in aluminum resistance. So we further characterize the subcellular localization of NIP1-2 when expressed in yeast as well as the native plant root cells. The NIP1-2 GFP recombinant protein, it was localized to the plasma membrane, suggesting that the protein is a plasma membrane localized protein. Yeah, further, we characterize their transport activity in the model yeast systems. What we found out is that the NIP1-2 expressing yeast cells facilitate the aluminum uptake activity when the medium was supplied with aluminum malate, but we not see any uptake activity with the aluminum ion or aluminum ion conjugated with different cellular ligands. This suggests, I mean, the aluminum malate is a transport substrate for NIP1-2.
Can you tell us what the cell type is there?
Pardon me?
Tell us what the cell type is? Is that the root cap?
So,
what is the cell type in,
this is
in the plant roots? What cell type is it?
cell type. This is the yeast.
What in the plant root cells?
This one?
Yeah.
This is the root cell. Yeah, root cell, root tip in the root tip region.
Is that the root cap?
Not root tip. So in 1 centimeter, so within the 1 centimeter of the root tip, it's not root cap. Yeah. So could be cortical cells, or I mean, yeah. That's it's an overexpression of 35S, so it's everywhere. I mean, we just want to see the general idea how the protein, where the protein is localized. Okay. And next, we use yeast system to further characterize the transport activity for NIP1-2. And this is the time course influx and efflux assay. As we can see, NIP1-2 facilitate net aluminum uptake activity and also net efflux activity for the yeast cell. These results suggest that NIP1-2 is a bidirectional aluminum malate transporter. Yeah, this slide summarizes how the systems work. And this showed a longitudinal section of the root section. So the left is the soil and the rhizosphere, and this part is the plant root cells. And the open area stands for the root apoplastic or root cell wall, and then circles indicate the plasma membranes of the epidermal, cortical, endodermal, pericycloparenchyma plasma membrane. Under low pH condition, aluminum ions are released from the soil, which can freely enter the root cell. So in the root cell wall, I would say, in the root cell wall, the aluminum ion directly activate the malate transporter or ALMT1, leading to the release of malate from the cytosol to the root cell wall, which can also be diffused to the rhizosphere. So in the rhizosphere, as well as in the root cell wall region, aluminum and malate can form a complex, aluminum malate complex. This complex is non-toxic to plants, plus they cannot enter the root. So that's how the exclusion mechanisms work. So previous people thought, oh, the malate and aluminum complex is non-toxic to plants. And plants has no way, to like spit out like animal, right? To push away the compound to the soil. So they thought that's not toxic. But in our case, we found that based on our mutant analysis, so this complex indeed are toxic to plants and need to be further removed from the root cell wall. And the root cell wall is one of the most sensitive target for aluminum toxicity. So this is achieved by the NIP1-2 transporter. So it removes aluminum malate from the root cell wall into the cytosol, and through the thin plastic flow, it can further move to the parenchyma cells, where it slowed proceed to the xylems, facilitated by NIP1-2. So this is an efflux activity. So it also fit for the bidirectional transport activity of this protein. So in this model, we see two components. One is ALMT1, the malate transporter. The other one is NIP1-2. So it's a aluminum transporter, right? So what's the advantage for the jointed function of ALMT1 and NIP1-2? So we know ALMT1 is the most important aluminum resistant genes in Arabidopsis. It accounts for 80% of the aluminum resistance in the species. So however, the malate release and spending, it costs carbon and energy, right? Malate is an important carbon source for the plants. So however, the NIP1-2 is a channel, and it just passively transport the substrates across the plasma membrane. It doesn't need energy. So with the addition of NIP1-2, we can see 15% increase in root growth under aluminum stress condition. So the coordinated function of ALMT1 and NIP1-2 can increase carbon use efficiency for aluminum resistance. So the next topic is Aquaporin and heavy metals, specifically with zinc. Zinc is an essential micronutrient for all plants. However, excess zinc is toxic to non-accumulating plants like Arabidopsis. The symptoms of zinc toxicity include stunting shoots, leaf chlorosis, root growth inhibitions. So mainly, the shoot is the primary target for zinc toxicity in Arabidopsis. So for plants to cope with the toxicity, so mainly involve the internal detoxification mechanisms, specifically like a zinc immobilization in the root. As I mentioned before, the general rule of thumb is like move the toxic metal ions away from the most sensitive part to the and keep in the non-sensitive part. And zinc toxicity target at the shoot. So plants need to keep the zinc in the root, which is less sensitive, right? And so the result is less zinc will be translocated to the shoots. The second important step is to transport the excess zinc inside the cell into the vacuole. So this is also a very important mechanisms for detoxification. So we have now, there are a few counterplus transporter responsible for zinc detoxification. So this including NTP1, NRAP3 and 4. So our question is, are there other transporter involved in this process? So we hypothesize that tip members of the Aquaporin family could be involved in zinc sequestration in Arabidopsis. So the rationale is that the counterplus transporters facilitate vacuolar zinc sequestration, and the members of the tip subfamilies are localized to the counterplus. So to test this hypothesis, again, we analyze mutant lines for individual members of the tip subfamily. And we found out that the TIP2-2 mutants are more sensitive to elevated zinc concentration compared with the wild type. Also in this slide, I include several very known mutants. They are hypersensitive to zinc toxicity, including CAT2-1 and CAT1-3 and TP1. So this suggests, the wild type function of TIP2-2 is involved in zinc detoxification. Next, we study the zinc distribution in wild type and mutant plants. We look at the zinc content in the root, shoot, and xylem sap, as well as leaf trichomes. So as we can see under the standard condition, so 2 micromolar zinc, there's no difference in root in zinc concentration for the wild type and the mutant plants in all tissue or samples examined. However, under elevated zinc concentration, here it's like a 60 micromolar in hydroponic solution, we can see significantly lowered zinc content in the mutant plants compared to the wild type. But in contrast, we see higher concentration of zinc in the shoots and the xylem sap. So this results indicate that the mutant plants could not retain or defect in the retention of high level zinc in the root, so that more toxic zinc is translocated to the shoot tissue, so here. And also we measure the zinc content in leaf trichomes. Surprisingly, and the mutant contains less zinc content in the trichome. Trichome is also an important organ for heavy metal detoxification. So it's considered as external organ for plant. So this result indicate that TIP2-2 facilitate zinc immobilization in the root and sequestration in the trichome. Then we further look into the distribution of zinc in the root cells. So we know the mutant plants retain less zinc in the root, right? But it's because less zinc concentration in the root cell wall or in the root cell, everything excludes the root cell, right? The root cell wall. So here is the zinc concentration in the root cell wall and root cell sap. Root cell sap is everything except for the root cell wall. So what we can see is for the root cell wall, we cannot see difference between the wild type and the mutant, but the mutant contains significantly less zinc concentration in the root cell set. This result indicates that the TIP-2 facilitates zinc transport into the root cell, but not just keep them in the root cell wall. So TIP-2 expression, it's induced by elevated zinc concentration as in both root and shoots. And amazingly, the expression is suppressed by zinc depletion condition, as shown here, the time course TIP-2 expression. So we see significantly reduce the level. So with the as a function of time treatment of zinc depletion. So this result indicates that TIP-2 expression is determined by zinc status of the plant. So we next take the expression pattern for mRNA and also for the subcellular localization of the protein. So we see the gas activity mainly localized on the root region. So that's from the root elongation to mature root region, and also the protein is localized to the plasma membrane because it's the TIP-2 GFP fluorescent is overlapped with a very well-known tonoplast marker, TIP-1-1. So this for linked with the RFP. So we further characterize TIP-2 function in the model yeast systems. So before I proceed, I would like to introduce the 4 primary zinc transporter in yeast. So there are two of them are localized on the plasma membrane. So that's ZR1, ZR2. So encoded for high and low affinity zinc transporter, and also 2 zinc transporters on the tonoplast. So that's ZR1 and COT1. So the ZR1 and COT1 facilitate zinc sequestration from the cytosol to the vacuole. So if these 2 critical transporters on the tonoplast is deleted, what happened under elevated zinc concentration? So what we can see, the zinc will be taken up by the plasma membrane localized ZR1 and ZR2, and we'll see the accumulation of zinc in the cytosol, but the cell lack the function for sequestration of the toxic zinc into the vacuole. As a result, the yeast cell would be extremely sensitive to elevated zinc concentration. So we can use this system to test function for TIP-2. So our prediction is that if TIP-2 is a zinc protein and it's localized to the tonoplast, so it will be rescue or complement or partially for the hypersensitivity phenotype of the ZR1 COT1 double mutant under the zinc toxicity condition. So we express a TIP-2 GFP fusion protein in the ZR1 COT1 background, what we can see here. So this fusion protein is localized to the tonoplast. Yeah, and then we test the zinc resistant phenotype. So this is the Zirc1, CART1 mutant with an empty vector or expressing TYP2-2. We can see under the standard condition, so the growth of both lines cannot be distinguished. So, however, under elevated zinc concentration, 60 micromolar zinc is not too high, but you can see the double mutants with the vectors show very strong growth inhibition. But the east line expression TYP2-2 shows partial complementation for the mutant phenotype. So yeah, okay. So next we treat the cell with BSO. BSO is a potent GSH biosynthesis inhibitors. So with these inhibitors, we see the elimination of the partial complementation of the TYP2-2 lines. So these results, yeah, see there's no difference compared with this one. This result suggests that GSH biosynthesis is required for TYP2-2 facilitated zinc transport and sequestration to the vacuole of the yeast cell. So in summary of this plot, so our results indicate that, or suggest that TYP2-2 facilitate zinc sequestration to the vacuole of the root cells, and also mediate the zinc immobilization in the root so that less zinc is translocated from the root to shoot tissues. So in conclusion, TYP2-2 is a critical player in zinc resistance and detoxification in Arabidopsis. So the last part of this section is Aquaporin and other heavy metal toxicity. So I don't have much time left for this project. It's still going on, and just show you several photos indicating that. So we identify some mutants for the in the Aquaporin family that are hypersensitive to cadmium and also to nickel. Nickel is also essential micronutrient for plants. Yeah. So more work is still going on. So in summary for my talk, aquaporin are generally believed to facilitate transporting polar but non-charged small solids and metalloids. So our findings provide several lines of evidence indicating that some aquaporin members could involve in metal ion transport and resistance. So further studies are required to elucidate the nature and function of the ion conducting aquaporins. So last slides, I would like to give a quick summary of some of other ongoing research in my laboratory. So first one is the aluminum resistant project with the 2 resistant genes in the wheat and normal aluminum resistant mechanisms in maize. So I have gave a plant breeding seminar in early spring of this year. So this is the YouTube link. So in this video, there's more detail about these 2 projects. So and also we have stop one. It's a mutant hypersensitive to low pH. So we've identified several. I mean, that's many suppressors. So this is a good project to study the mechanisms underlying low pH stresses. And also we are involved in identifying or elucidate the plant hormone and their roles in plants resistant to low pH stresses. So I think this all could be good opportunity for graduate students and postdoc also. So with this, this end of my talk, I would like to thank my current and former lab members and also my collaborators listed here. So I only have a few minutes left. So I would like to stop here and thank you. And thank you all for your attention. And I would like to answer questions. Yeah.
Do aluminum and zinc permeate the membrane in a hydrated form?
I cannot hear.
Zinc and aluminum permeate the membrane in a hydrated form or a dehydrated form?
Yeah, so this is, I know your question underlying, I mean, so this is very complex question. I mean, it's very difficult to study the chemical form of like aluminum and zinc, I mean, inside the cells. But I can say, I mean, the aquaporin, we have many circumstantial evidence. So for instance, zinc, it's for zinc GSH complex. So it's probably a neutral charge, but we need more direct evidence to make the conclusion. Yeah.
Let me ask you one more question. Do you think aquaporin is a misnomer? It has been a misnomer from the very beginning when Martin Chris Beals used it to describe water changes in plants.
So I didn't get your.
Oh, do you think the function that the natural selection has given that protein that's called aquaporin a selective advantage for transporting water, or is its real function for transporting other kinds of things?
Oh yeah, I mean, this is a great question. So there are so many members of aquaporin. So there's many studies indicates besides water, so aquaporin can also transport many like small solids, including like carbon dioxide, ammonia, boric acid, right? So that is many, and still many aquaporin members functions are not clear. So only a few of them is being characterized. So that's very diverse. I mean, that's.
So one thing that's interesting, almost everything you mentioned will not permeate through a lipid membrane, through the lipid part of the membrane. It's a transport protein. Water, on the other hand, will permeate through a lipid membrane.
But very limited.
Conductance is small, the area is huge. So the lipid membrane actually transports probably half of the water. So the selective pressure for water movement would be minute for aquaporins.
Yeah, I mean, this is a good question. I mean, that's not still, the mechanisms are not very clear. I mean, for aquaporin, there's two constriction regions. So that one is MPA constriction region and also AR-R constriction region or selective filters. That people thought, I mean, that's critical for solute selectivity. But obviously, I mean, this is not the only component that determine the substrate selectivity. So there's still many unknown questions left for aquaporin. Yeah, sorry.
I was curious about tip 2-2. Is it well conserved in other species? Does it have like clear orthologs, or is it part of a large gene family?
Yeah, I cannot hear very clear with your mask.
I'm curious about tip 2-2 and whether it's conserved at the level of orthology in other species. And I'm curious because we have a rootstock we work with that protects the scion from accumulating heavy metals, including zinc. So If you
Oh, yes. I mean, that's definitely a good question. So my answers are, we don't know yet. I mean, we just look at in Arabidopsis, but haven't looked at other species. But I can give you another sample. It's like NIP. I mean, that's for aluminum. So it was we identified in Arabidopsis, but now we are working in the weed. So definitely, I mean, that's the mechanisms. It's conserved between the monocotyl and dicotyl species. So we are hoping using this knowledge obtained from Arabidopsis to improve aluminum resistance and yield for the weed. Yeah. So yeah, I guess it's the answer, but I have no evidence yet. Yeah.
So I have a question about the NIPs. So can you hear me?
It's yeah. You speak loud with the mass. I mean, that's always.
Yeah, you just said the NIPs 1-2 is a bidirectional transporter.
Right.
So I don't know if the direction is depend on the position, of the cell, or just concentration of the concentration of the material.
Yeah, I mean, this very common for the NIP members. They are function as a bidirectional transporters. So there's many other evidence. So for other NIP members, so for instance, for the arsenic transporter, so it's also facilitated by a few members in the NIP family. So they're all bidirectional. So maybe it's a common feature for this gene family.
Yeah, I mean, according to one of your slides, it shows the direction, the aluminum malate compound basically transport from out, from the environment inside the epidermis cell, and then just go all the way to the xylem, right? So this is one direction. So is there any possible that the aluminum just go with outside from the xylem to the environment? If the concentration of the aluminum in the environment is relatively low or something like that, because it's bidirectional transportation, right? So
yes, I mean the NIP1-2 or other channel proteins, they are passive channel. I mean transport substrates based on the concentration gradient. So it's just like a pass, I mean, like a gate. I mean, it's not an active. So I think that's, I mean, this is the factor you need to keep in mind. Maybe the root, epidermal cortex, they are closer to the rhizosphere. So that has more chance for the aluminum get into, so they have higher concentration that can, so these all down the concentration gradient. So that's what.
all right, thanks.
Okay, yeah. Hi, Ross.
So Yale. So for the 2-2 transporter, you talked about facilitation of zinc from the cell wall to the cytosol, right?
Mm-hmm.
So presumably you're talking about freely mobile zinc, not in complexation with the cell wall. But the wall itself, the polysaccharides, can complex very large amounts. So do you incorporate that into your analysis?
Yeah, I'm very sorry. I cannot hear very clearly.
Oh, really, Yale?
Oh, yeah, that's you.
So
That's what I asked to wear a sheet instead of a mask, because the previous seminar got a lot of complaints about, yeah. So if you can, or I can go closer so that.
I can speak really loudly.
Okay.
So I'm asking, do you know how much of the aluminum or zinc is complex into the cell wall itself? So it's not freely mobile, it's complex with pectins, the polysaccharides. So it's bound.
Mm-hmm.
Does that vary very much in your analyses?
Okay, so I don't know exactly how much, but I can show you back maybe probably. Yeah, I mean, the one, we just measure the zinc content in the cell wall. versus the cell sap. That's all we have now. That's all the knowledge we have. Yeah.
Yeah, so I think the amount that can be bound polysaccharides and the amount that's freely mobile can vary a lot. So that would be interesting to
Yeah.
compare the apoplastic fluid with the cell wall bound components.
Yeah. Yeah, that could be. Yeah, definitely. Yeah, agree.
There's one question.
One
in
more
chat.
question. Yeah. Oh, one more.
I also want to refer to your model about NIP.
Yeah, speed up.
So your model about NIP1 and aluminum resistance. Your model has how said it moves. So you move the aluminum malate from outside to the inside. Have you seen that NIP1 is localized polarly in any type of cell, or is it just everywhere on the membrane?
Oh, you mean the gene protein localization or expression?
Yeah,
Yes, it's in the root tip. I mean, that's we measure. I mean, examine the expression pattern for both NIP1-2 and ALMT. You mean? I mean, that's the two like a partner. So their expression patterns overlap. So this quite consistent with their function. They work coordinated. I mean, actually, the ALMT1, it's epistatic to NIP1-2. We have independent study to indicate that. So yeah, I mean, I would say, I mean, their expression patterns in the same position of the root. So I don't know if I answered your question.
Well, my question is whether it's polar, like whether it localizes to one part of the cell more than the other part.
One part of the cell wall. I think that's the you mean, I mean, so there's a Casparian, right, in the root region. I think that's everything outside the cell wall. As long as there's a NIP1-2 expression, I mean, it will like move the aluminum malate complex from the cell wall into the cytosol. Yeah.
Time for one or two more questions from Zoom.
Okay. Yeah.
There's one, what happens to the aluminum zinc after it's transported to the vacuoles? What do you think GSH contributes to this?
We have aluminum deposit in. Yeah, let me say thank you for the enter the silent. Okay, good. Yeah, so after loading into the silent, it will move to the chutes based on the respiratory stream, right? I mean, that's all like water and many other solute are translocated in this way. Yeah, I hope I answered your question. I mean, that's all go everything with others.
All right, let's all thank Jiping again for a great seminar.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.